Alrighty, here we're talking about the biodegradation of plastics, and specifically we're talking about the enzymatic biodegradation of plastics. And this process is kind of a new technique or it's new research that's been ongoing. And we found a couple different methods of degrading plastics. There's only a few select enzymes that can do it and a few select plastics that can actually undergo this process. And we'll touch on those shortly in this presentation. And it's also important to note that we are using microorganisms to create these enzymes, and then these enzymes are going ahead and degrading plastics. So before we start, or before we get further into this, we're going to talk about today's plastic crisis, just a brief overview. We've seen a 21% increase in plastic production from 2014, and that puts currently the global plastic production at 390 megatons. And the US is actually responsible for 42 out of that 390 megatons, which is a significant percent of the global plastic waste generation. And we also can see here that 83% of plastic actually goes to waste, with as little as 15% of what is recycled actually being, that is actually reused more than once. So we barely see any plastic actually being recycled, which is pretty sad. So, and that is kind of a product of the linear consumption model. And we can see that below in this figure where resources are extracted, particularly petroleum-based products, which we use to make our plastics. And then we produce them into these plastic products where they're distributed, consumed, and subsequently disposed and placed in the landfill. And this whole cycle is, we want to break free of this linear model ideally, but there's a lot of difficulty with producing biodegradable plastics. And currently there's a very small percent of global plastic production that is biodegradable. So to kind of combat this problem, we're going to look at the biodegradation of plastics. And let's take a look at some of our common plastics today. And we can also note that these plastics are not biodegradable, which is kind of an issue when it comes to getting rid of them in a environmentally friendly way. And we'll see why we want to transition into enzymatic degradation of these plastics as a solution. So touching on some of the most common ones, we see polyethylene, which is in number 2 and number 4 plastic, high density polyethylene and low density polyethylene. And we can also see polypropylene, which is number 5, and polyvinyl chloride, number 3, and polyethylene tetraphthalate, which is number 1 plastic commonly seen in water bottles. And then we also have polyurethane and polystyrene, but some of the more degradable plastics are actually polyurethane and polyethylene tetraphthalate. And we're going to look at some of the methods that we can biodegrade these with. But like I mentioned earlier, because these plastics, these plastics are not, they're not all biodegradable. There's only a select few that are, but also that most of these plastics are extremely difficult to degrade unless you have some sort of enzymatic push. And we're going to talk about that very soon. So you can see that, so the solution is to just use organisms that produce enzymes that are capable of degrading these plastics, because if they're just in nature, there's no way any of these will be able to get degraded. Now that's why they're sitting in landfills and filling up landfills currently. But here's a quick example on the left, bottom left here, of an organism, and it can produce an enzyme, and that enzyme is capable of degrading a plastic. And this is here polyethylene terephthalate here, and it produces monomers, which then can be recycled or these monomers can be made into more valuable products, which is called upcycling. And you can see on the right here, this would be a bunch of organisms on a piece of plastic, and they're producing enzymes that are degrading that plastic. So just briefly to look at the entire cycle here, we can see that we're taking mixed plastic waste and we're placing it into some sort of enclosure. And at the top, we're producing enzymes. This is supposed to say enzyme production. And these enzymes are produced by specific organisms and they're inserted into this mixture. Here, the enzymes are able to decompose or degrade some of these plastics into products, many monomers as well. And now this mixture is then separated where the toxins are removed, and then the secreted products and the biomass products are separated as well. And then we have some non-biodegradable products left over after this process. And these are then subsequently treated with physiochemical treatment and then reinserted into the system to get further degraded. So this is kind of an overview of that process, and it's kind of an upcycling process where we are aiming to produce more valuable products out of our plastic waste.
Alrighty, so I think we'd like to introduce now this new project called the Mix-Up Project. And yes, it's for the motivation of plastic upcycling. What does that really mean? That means that it's going to motivate a bunch of scientific and engineering work. Already, by now, 2023, it's produced a bunch of publications in literature, various journals. Okay, great. So why do we need to go through all this effort and do all this work just to have organisms break down stuff? Isn't that just what organisms do, right? Well, that depends on what the stuff is that you want to break. So carbon backbones are difficult. If you think of polyethylene, it's just a straight chain of carbon, like tens of thousands of, atoms long. Great. How do you start cutting that up? Typically, you would want a functional group in there. What's a functional group? That's, like amine, amide, carboxylic acid, ether, ester. Sort of the stuff that you might remember from Introduction to Organic Chemistry, right? Biological catalysts are referred to as enzymes, and these enzymes are not magic. When you want to hydrolyze something, these enzymes are typically going to require some functional group, to break. So you can see sort of at the bottom here, we have this polyurethane segment, and we can see here this sort of functional group in between the C2H4 and the CH2,4 and the CH2,6, right? We've got this big functional group. That's something that an enzyme can cleave. We can see this PET section, polyethylene terephthalate, if you're curious. And again, this has functional groups. So, okay, we know what we can break apart. How do we break it apart biologically? Start with polyurethane. Polyurethanes. So fungi are filamentous. They're these creatures that are essentially designed to grow very close to something to, I guess, penetrate its pores with mycelium. And what it does is, you can think of it almost like a sea cucumber. It excretes degradation enzymes and then is able to digest stuff. Okay, so fungi is good at digesting stuff, breaking down stuff that typically other organisms find hard to break down. Great. What does that have to do with polyurethane? Well, we have this genus of fungi called the Cladosporum genus, and pretty much what a bunch of scientists did is they cast this big old plate of polyurethane, slapped a bunch of this fungus on there, and then left it for a month. And you can see in this diagram right here, recorded the weight loss of the plate. So the weight loss of the plate is, this is how much plastic has gotten degraded. We can see that with the plate as the, single carbon source, it loses like a third of its weight. That's pretty significant. And even more significant is that we have a phenomena called chain cleavage. And all that means is that it's not eating it from the ends. It's actually breaking up this polymer in the middle. Okay, that's pretty interesting. What about the other thing I was mentioning earlier? What about PET? All right. You can use a couple things for PET. You can use cutinases and you can use PET hydrolases. So a PET hydrolase is really self-explanatory. It hydrolyzes PET. But why did I mention cutinase? Have you ever picked up a leaf and, maybe scratched the waxy surface, right? What is that called? That's called the cuticle. Cutinases are enzymes built to break down waxy cuticles. So this is an enzyme that is relatively prolific in nature because it exists to break down a compound that's very common in nature, this waxy cuticle substance. So you can see in this top diagram here that we have some cutinase that can hydrolyze this PET. And we can see in this bottom diagram what, like a PET hydrolase would do at the very bottom where it's labeled PET. This is remarkably similar. And so we have this class of common enzyme that we can work with. Okay, that's great. How do we work with that? Well, there's a lot of different ways, but it seems that the way these EU mix-up groups tend to be working on is they have some media that has a large proportion of insert thing you want to break up here. You inoculate it with a bunch of whatever organism you want to break, plastic X down. And then you let it sit for a while, take it out, and then slap it on a new media with a streak to isolation, and you get different colonies. And hey, which one of these colonies grows the most on this polymer X? And then you choose that one to study. I think that's probably all we want to talk about for today.